Okay, everyone, thank you for coming. I'm Jen Flannery, for those of you that don't know me. I used to be here as a student in the Paleo Lab, graduated in 2008, and now I'm over at USGS. I'm really honored and happy to be hosting Dr. Kristine DeLong today. Kristine really loves USF. All three of her degrees come from USF. She has a bachelor's from the Department of Engineering, and then a master's and PhD from here, College of Marine Science, from the Paleo Lab. Since she graduated from the College of Marine Science, she did a postdoc over at the USGS and is now a professor at the Louisiana State University. So I hope you all enjoy Kristine's talk on reconstructing tropical climate variability from massive corals. So, thank you today.
Thanks, Jen, and thank you for having me come over and host. So it's always nice to come back to Florida, see friends and colleagues and family. So today I'm going to talk mostly about work that I've been doing since I left USF, work that I'm doing in collaboration with the U.S. Geological Survey over at the Dry Tortugas. So, and my beautiful picture here, for those of you who haven't got out to the Dry Tortugas, this is Dry Tortugas with Loggerhead Key with the lighthouse, and then Fort Jefferson over here in the background. Hopefully, especially students, if you get a chance to go out to Dry Tortugas, hopefully you'll get a chance to go out there and see the beautiful coral reefs. Also want to thank my collaborators. So Terry Quinn, who's now at the University of Texas, Dick Poor, Jen Flannery, Chris Rich, over at the USGS, Ethan Goddard here at USF, Tony Greco, and then also my colleagues in Taiwan, so River Shin, he does my uranium thorium dating for me. Also IODP, I've worked with them in the past, the National Park Service, so lots of different people go into doing this type of high resolution paleoclimate reconstruction, so I want to thank all my collaborators. So kind of start off with an overview of what is my interest in research, and that's looking at climate in the tropics. So the tropics have these very large warm bodies of water. So here in the Western Pacific, then also here in the Gulf of Mexico and Caribbean. So my dissertation work was focused here in the South Pacific. I'm not going to talk about that today. I'm going to focus more on the Gulf of Mexico, but I do have research interest in both regions. Now, when we start to talk about climate variability in the tropics or anywhere, one of the limiting factors is the instrumental record is just too short. So we don't have good records. So how do we bridge that gap to see what was climate like in the past? So we can use climate records such as tree rings or varve sediment cores or ice cores. And in the tropics, we use corals. Now, as I mentioned previously, we have this limitation with the instrumental record. So this is a figure, actually I borrowed this from my dissertation, to kind of illustrate where this lack of information is. So we have a database called COADS, and this is all ship of opportunity recordings of sea surface temperature, and it all goes into this big database. So these gridded data sets, such as the Hadley data set, or the Extended Reconstruction SST data set, they all use this COADS data set. And these are all observations that are mostly along the coastlines. But if you look at our tropical regions, you'll see there's very few observations here in the Pacific, and then over here in the Caribbean, Western Atlantic, and into the Gulf of Mexico. So very few observations when we get into the early part of the 20th century. As we move forward, we have more and more observations. So how do we fill in this observational gap? We can use paleoclimate reconstructions. There's all kinds of different proxies that are available. But each of these proxies have certain things that are required so that we can do a good reconstruction. So first off is location. So just like in real estate, we need to have a reconstruction in the right location. Okay? We also need to have a way to establish time or chronology in the record. We also need to have some sort of resolution. So what is that sample-to-sample time step? We also want to have a proxy record that's sensitive to some climate variable, whether it's temperature or precipitation. Okay? The next step is we need to calibrate that proxy. So if we're talking about tree ring records, we're looking at variations in growth widths from year to year. So we need to calibrate that to a precipitation record. So we're going to calibrate the proxies. The last step is the error in that reconstruction. So think of this just if you had an instrument, we were going out into the bay to take a measurement. That instrument has a precision. We need to understand what that precision or the error of our reconstruction is. So these are all things to consider. So now I'm going to talk mostly about corals as a paleoclimate proxy. Okay? And I'm going to give you a little background on corals so you understand how we do this. So the image over on the left is a cross-section of a coral, and the top is the live animal. Below that is the skeleton. Now the way the coral grows is it puts up sidewalls and then a floor, lifts itself up, puts up more sidewalls and a floor. So it's kind of like building a skyscraper. So if you look at the coral polyp, you see the walls below that and then the floors coming in below. Ooh, sorry. Hit the wrong button. Okay, so as the coral grows, it lifts up, so you get this time chronology building up within the skeleton. So when we go out and sample a coral, so this is a large, massive coral from the Pacific, and you'll see the diver at the top with a drilling rig. We're going to sample the colony through the vertical axis. So we assume this vertical axis is the maximum growth axis. So we want to get the longest time record possible. So from the center of this massive coral colony, we're going to extract these cylindrical cores. From the cores, we're going to take slabs out of those and expose them to an X-ray. Inside the X-ray, we can see annual density bands. This is how we're going to establish chronology. So if we know the date that we collected the core, so this particular one is 1992, we can count those annual density bands back through time to establish our chronology. Next step is to have some proxy within the skeleton. So I'm going to look at chemistry variations within the skeleton, and that's strontium calcium. So here you'll see these red, blue, and green lines. These are all sampling paths within the skeleton. Okay. All right. So the research question I'm going to focus on today, we're going to talk about the Gulf of Mexico. So one of my former lab mates, Julie Ritchie, her master's work was looking at Pygmy Basin. Oops, sorry. And she has a very nice high sediment rate marine sediment core where she looked at the G. rubers. And inside this record, she sees quite a bit of multi-decadal scale variability within the Gulf of Mexico. And if you look at the little ice age interval, her record suggests that the Gulf cooled up to 3 degrees throughout the Little Ice Age. So this is a lot of cooling. So one of the things we wanted to look at is, can a high-resolution record provide us more details on what's going on in the Gulf of Mexico? So if we look at the instrumental records, so the second plot here, I've pulled the grid box for Dry Tortugas, which is a study site from the COADS database. This is the number of observations. So you can see here from 1940 to present, there's quite a few observations. And then it starts to decrease. When we get into the 1920s, there's very few observations. So this shows us we just don't know a lot about what's happening in the Gulf of Mexico as we get back further in time. So it's not even the whole 20th century. It's early part of the 20th century. We really don't have good data. So our research question is, can we produce a high-resolution sea surface temperature reconstruction to tell us more about what's going on in the Gulf of Mexico, and particularly in the Little Ice Age interval on interannual to centennial timescales? Okay. Now, another consideration in my research, and this is with the Dry Tortugas, any location that I've been working in, is to take into account recommendations that have been made to the community. So this is a particular paper I'm citing by Jones et al. From 2009. And they're making recommendations for high-resolution paleoclimate reconstructions. So Three of the key things they recommend for coral reconstructions is replication. Typically, this is not done with corals because one, corals are a species that is under stress, so we don't want to overstress them by taking too many cores, so typically we don't replicate. Okay? However, if you think about tree ring chronologies, they take 2 cores from every tree, and that's to reduce the biological variability. Okay? So having replication lets you reduce that error. Okay? The next thing is to extend the record back further in time. Most of the reconstructions that have been done have all been done with live corals. So the next step is to go in and take corals that are already dead or subfossil corals to extend the record back further in time. Next thing they recommend is to calibrate with long local records of SST or of temperature. Okay? Typically this is hard to do as well because a lot of tropical locations do not have local sea surface temperature records. So we use gridded data products. Okay? The other thing is long records. They're just not out there for a lot of locations. However, that being said, with the dry tortuga study that I'm going to show you today, I've addressed all 3 of these concerns outlined in the Jones paper. Okay, so this is dry tortugas, my study site. So we're here in the Gulf of Mexico. So dry tortugas is 100 kilometers west of Key West. So up here in the northern part of the Gulf of Mexico is Pygmy Basin, where Julie's record is located. Then I've also put Key West here just for reference. So the dry tortugas, this is an ideal place for this study because there's 2 buoys out here that have been collecting hourly temperature records since 1992. So dry F1 is over here by Fort Jefferson. Fortunately, we lost this buoy back in 2005 with the hurricanes. It was replaced later in 2007 with the buoy up at Pulaski Shoals. Oops, sorry. So I'm going to take these 2 SST records, combine them together into one record. So the coral sites are up here by Pulaski Reef. So there's 2 corals I'm going to show you data from today, the 08 PSA and F cores. Then also here just south of Fort Jefferson are 2 records that were collected back in 93 by the USGS. So 93 dry SS1. Chris Maupin published this work in his master's work. And then there's the dry SS2 coral, which is a new record that I'll show data from today. And this core is a subfossil coral. It does not have a live top. I also want to point out the location here of Loggerhead Key and the Carnegie Marine Laboratory. So I'm going to show you some data from this site as well. Okay, so we'll move forward. All right, so just a little background on the corals. So we went out in 2008, the USGS, and we cored several coral colonies out there. So this is the largest of the colonies. So 08 PSA, it's a Cedastria sedaria coral. These corals typically grow very slow, so about 4 millimeters a year. So a tall colony is such as this. So this is almost 3 meters tall, or sorry, 2.5 meters tall, is an older colony. This particular colony was dead on top. So you can see there's fire coral growing in the top. So that central vertical axis, we took 1 core from the center there, but this core is going to have an unknown date at the top. So to bridge this area where we have missing coral, we took a second core from off the side through the live portion of the colony. So this is the 2 cores collected from the 08 PSA colony. So one with the live top, so you see this is a very long core going back through time, and then the second with the dead top. So we also sampled a second smaller colony, so the 08 PS F1 of the same species. So we have the cores and the x-rays from this colony as well. So this colony has 2 sample paths throughout most of the record, so we've replicated within the colony as well. And in this part of the longer coral record, we've replicated through certain intervals as well. Our third colony we sampled is that 93 dry SS2 core that has a dead top, and you can see at the top there's some bioerosion at the top. So that's the second record, or the third colony we're going to look at. Okay. So taking these records together, so we have cores where we have unknown dates at the top. So to establish a date, I used two different methods. So one was cross-dating, and this I borrowed from tree ring research, where we're going to take the variations in the chemistry of the skeletons and line up those variations. So when the variations line up, you can piece a core with a live known date with the core with the unknown date. The other way we're going to establish a date is uranium-thorium dating. So I have several dates we collected. So from the top of the dead core from the large colony, and then also several dates from the top, middle, and bottom of the 93 dry SS core. So the plot I'm showing here is that strontium-calcium annual density band cross-dating chronology, and on the y-axis is uranium-thorium age. So, and I'm showing for each of these samples, we have the bands that we counted within. So we're going to have the error for the uranium-thorium date, and then also the error of our cross-dated chronology. So you can see here for all four of these uranium-thorium samples, they line up within analytical error with the uranium-thorium dates. So this tells me that my cross-dating of the strontium-calcium and density band counting is confirmed with the uranium-thorium dates. So the next thing we want to look at in corals, especially because I have cores with dead tops, so we want to make sure that the mineralogy is pristine, that we don't have any diagenesis. So Jen and I actually did these with Tony Greco here at USF. So we have samples from the top of the dead core from the large colony, and then also samples from the top, middle, and bottom of the subfossil coral collected in 93. So in all three of these corals, we don't see any indications of diagenesis, till we get to the very bottom of the large part, or the large colony, the very bottom of this coral. So here you can see in the x-rays that the colors become quite lighter, so that's an indication that the density is becoming larger within the bottom of the colony. And inside the SEMs, you see the pore space, but now we have secondary aragonite crystals that are forming within those pore spaces. So this tells me that the bottom part of this coral is altered. So we're not going to include this bottom section in our final reconstruction. So next thing I wanted to test is, remember at the beginning I mentioned we always sample along the vertical axis. And this is something that's been done with coral reconstructions now for 30 years. However, I've started to notice that maybe we can go off the main axis and still have a good record. So what I wanted to test with the large colony, since we have these two cores, is the annual density bands line up between the cores. So if I have samples here that are almost vertical, compare them with 45 degrees off vertical and 70 degrees off vertical, do I get the same chemistry? Okay, so that's one test I wanted to look at. This is particularly important for this 93 dry SS coral, because you'll notice as we go down core, the angle of the growth is changing. So when we get to the bottom, it's almost horizontal. So if I'm going to use this coral to extend the record, I need to be sure that I don't have any shifts occurring because my growth angle is not vertical, it's now almost horizontal. So I had one of my undergraduate students actually sampled and conducted this test for me. So this is the results of that test. So we have the red lines are the samples from the almost vertical path from the dead top core, and then our 45 degree is the gray lines, and then the black lines are the 70 degrees. And then for comparison, I've also plotted the green lines from the dry SS2 coral. So in this section, they're almost vertical as well. So here we do not see any mean shifts between the four paths, and that variability is within analytical precision for all 4 of these paths. So this gives me confidence that I can use sampling paths that are off vertical throughout my reconstruction. So if there was a problem with this, and it's been reported with some of the columnar corals from the Pacific, we would see a mean shift. So we wouldn't just see the same seasonal cycle, we would actually see a lot higher strontium calcium values. So here we don't see any shifts with respect to coral colony orientation. Okay, so these are the strontium calcium results from the 4 cores. So the chemical proxy I'm using is strontium. It substitutes for calcium in the coral aragonite skeleton. So this substitution has an inverse relationship with temperature. So the sampling resolution I'm using is monthly. So between each of those annual density bands, I'm extracting about 12 samples a year. So another thing to point out, this is a group project. I did not sample everything here. The A1 and the A2 were sampled by myself. The F1 and the 92 dry SS2, these were sampled by Chris Maupin over at University of Texas. So both of us similarly trained, but we did not talk to each other or compare results as we went through this process. So only at the end, once we ran all the samples, did we compare. So it's kind of a semi-blind comparison between the 2 records. So if we look at all 4 of the records, we do not see a shift as far as the means between colonies. Remember, these are colonies growing next to each other on the reef and at either end of the dry Tortuga, so 10 kilometers apart. So we don't see shifts in variance. We don't see shifts in the seasonal amplitudes. If we look at the average RMS difference between the colonies, it's about 0.2 millimoles per mole. So our analytical precision, 2 sigma, is 0.26. So my differences are less than analytical precision. So it's within the noise of how well we can measure strontium calcium. Okay, so I know there's a lot of data points up here. It's a lot of work. So I'm going to rework the data and show you reproducibility in a different way. So what I'm plotting here is taking the seasonal cycle out of each of these cores. So we're looking at anomalies. And then additionally, I'm going to take a 36-month smoother through the data. Okay? And in the reconstruction, we're not really interested in the seasonal cycle per se. We're interested in that long-term variability. So we're going to take out everything that's below 3 years. The next thing I've done is I've plotted each of the records as a shaded area that represents a 2 sigma analytical precision. So everywhere where these shaded areas are overlapping, those differences are within analytical precision. Where they do not overlap, those differences are larger. Okay? So if we look at the whole record, 98.9% of all of the strontium calcium variations are within analytical precision. So that's very good. It makes me happy for this reconstruction. If we look at the correlation, it's quite high. So our R-squared values are above 0.8 for monthly. And then for 36-month smooth, they vary between 0.74 down to 0.10. So again, this tells me we do have shared variance between the records. So at this point, I'm confident that these 4 different cores from either ends of the parks, different size colonies, different growth orientations, they're all giving me a similar record. So now we're going to take that record and compile and master strontium calcium chronology. And that's the black line you see here. Okay. So now that we have our record compiled and together, next we need to do a calibration. So for this calibration, I'm going to use those 2 sites. So here within the national park. So I've combined those 2 records together into one sea surface temperature record. Okay. Now I'm also going to take a record from Sand Key. So this is a coral reef site just south of Key West, Florida. So the reason to use Sand Key, if you look at the dry Tortugas, which is in black, you'll notice there's gaps in the data. So there's also periods where we don't have observations. The Sand Key record is a little longer, and it doesn't have as many gaps. Okay? So with both of these temperature records, I'm going to perform a calibration of our coral strontium calcium to sea surface temperature. So shown here is the black dots are the strontium calcium with the SST with their error bars. Since I have 4 records that I'm averaging together, I can calculate error for every strontium calcium observation. Since I have hourly SST, I can also calculate error for each of those monthly averages. So all of that information goes into my weighted regression. Okay? So once we do that regression, we get a slope. I've plotted I've given the calibration with strontium calcium. This is typically how it's shown in paleoclimate reconstructions. However, the sea surface temperature equal sign is the probably correct way to show this, since that's what we're reconstructing. So I've given the equation both ways. It gives the same answer. If we look at the error of regression, so this is the difference between our reconstruction with each of our sea surface temperature data points, I can assess what my error is, and that's 0.67. So our analytical precision error in temperature space is 0.5. So we're very close to our analytical precision with our calibration. Okay? Now, the next thing we want to do is we've calibrated with local SST and then also with another temperature data set that's in Key West. We also want to assess that reconstruction for another interval. Oh, sorry. Forgot I had this flying in. Another thing that I've been working on is with the replication studies is how many corals do we need to sample? So in that Jones paper I mentioned at the very beginning, they suggest that every coral reconstruction needs to sample 3 colonies. But nobody's ever tested that. So I did test that with my dissertation work with Porites. I found that you only need 2 colonies with that species, and I'm doing the same thing with the Cydastria sedaria. So what I'm finding here is our error of regression is reduced by 52% by including 2 colonies. So if a single colony, our error is up over 1 degree Celsius. Okay? So the next thing we want to do is to verify our calibration for another time interval. So, and this is something when I plotted this up the first time, I did a little dance in my office. So hopefully you guys will think this is as exciting as I do. So what this is from the Carnegie Marine Lab. So that loggerhead key, remember the lighthouse in the very first slide I showed you? So that lighthouse has been out there since the 1800s. And there was a lighthouse keeper who went out every day and made a temperature measurement. Put a bucket in the ocean, put a thermometer in, write it down. So Vaughn back in 1918 compiled all of these records and published that. So we've taken that lighthouse keeper's record and compared that to our coral reconstruction. So the lighthouse keeper record is here in the black, and the coral record is in SST. Or I'm sorry, the coral SST is in red. Now a couple things to note. The first calibration I showed you, so when we measure the corals, strontium calcium going down core, that's all done based on length. So it's in millimeters. Okay? To get it into the time domain, I match up the coral strontium calcium to a time record. The time record I use is a temperature record. Okay? So I'm making the coral record fit the temperature record as far as time goes. Okay? For this particular verification interval, I did not do that. What I did is just assumed that every February was the coldest month and every August was the warmest month. Everything in between was linearly interpolated. So this is not matched up to the temperature record at all. Okay? And what's amazing is there is quite good agreement between these 2 records. So there may be some winters and summers that are off by a month. That's to be expected. I did not make them line up. Okay? If we look at the verification, so that's looking at the error between the 2, it's 0.86. Had I lined these up, this number would be smaller. Okay? So we have now temperature records from the lighthouse keeper from 1878 all the way up to 1908 with very good agreement. So R squared of 0.85. The other thing that's really interesting about this is the very bottom of the 1 core where I have a live date. The A2 and the F1 cores, these are the cores that I cross-dated and established the age with uranium thorium dates. So do you guys see why I did a little dance now? So like, this is really cool that it matches up. So no one's ever done a verification going back this far in time. So this tells me my coral strontium calcium calibration is stable on 130-year time scales. So that's a big deal. Okay. Now there are a couple little discrepancies here. So here we have 1 year of the lighthouse keepers data where it looks like something happened. They did not record the summer. So fortunately we have other lighthouse data from along the Keys. The other lighthouses do not report a summer missing. So something happened here with the recording. So I don't think this temperature record is correct here. So the other thing is we have a very cold year here in the coral record. This is the coldest winter in the whole record. It is duplicated in both cores. Again, these are two cores 10 kilometers apart. So I think this is real, but it's not captured in the temperature record. So there are two discrepancies within the verification interval. Okay, so now we move on to the full reconstruction. So the way I've plotted this, again, it's looking at strontium calcium temperature anomalies. So I've removed the seasonal cycle and I've smoothed that over 36 months. Here are the shaded areas. So there's a light gray and a dark gray. So this represents my regression errors that I calculated in the calibration interval. So this tells me how confident I am in the reconstruction. These regression errors are based on the number of cores, so similar to what they do with tree ring reconstructions. So where I have 2 cores or 3 cores, I have smaller errors. Where I have a single core, I have larger error bars. So here I have less confidence in this interval. Here I have more confidence. So one of the first things we wanted to do was compare our reconstruction to those gridded SST data sets that everyone uses, whether it's coral reconstructions or using climate studies. Everyone likes to use these gridded temperature data sets. When we compare that to our coral reconstruction, you'll see our R squared for monthly is high, 0.92, but if we look at monthly anomalies, it's quite low, so 0.12. If we look at the record, here from 1970 on, there's good agreement, but as we move back further in time, we have this cold interval here in the 50s and 70s that the Hadley data set does not pick up, and then there's a warm interval the coral doesn't show. So back here, all of this just doesn't agree as we expect. So at first you scratch your head like, ooh, something wrong with the coral, or is there something wrong with the Hadley? So we wanted to compare this to another data set. So, and this is just that same plot I showed from the beginning, showing the number of observations in the Hadley dropping off. So we do have another temperature record that's a long record available, and that's an air temperature record from Key West. And this goes back to 1986. So you'll notice here we've got 100 kilometers apart, air temperature, not water temperature, but we do have good agreement between these records. So R squared is a 0.86, and then for our monthly anomalies, smooth, 0.31. So we are capturing that similar variability, and the temperatures are moving in the same direction. So from this, I can assume that the coral reconstruction, the air temperature, we have the lighthouse keeper data. So all of this is telling me the coral record is giving me a good reconstruction. The Hadley data, I'm not so certain about as we get back into the early part of the 20th century. Okay? So what are some other things we see in this coral reconstruction? So we had Julie's record we talked about at the beginning, where she saw 3-degree swings in the Little Ice Age. So if we look at the Little Ice Age interval in this coral, so from 1934 to 1870, the temperatures here are 0.8 degrees C colder than average. But we do have swings as high as 2 degrees Celsius throughout this interval. So similar to what Julie was seeing in her record. So I plotted hers here on top with the green. So they're slightly shifted because the northern part of the Gulf of Mexico is just a little cooler than dry Tortugas. But you'll see the trends here between the records are quite similar. So, and these temperatures are plotted to the same scale. Okay. So next we want to kind of tease apart the record and see what other things we can learn from our reconstruction. So here I've taken those summer extremes and winter extremes and plotted those separately. So the top is the summer. And if we look at just the whole interval, so 274 years, we see we have about a 1 degree Celsius warming. If we look at the winters, it's only 0.36, which is kind of interesting when you think about it, that we're seeing the warming mostly in the summer temperatures, because we kind of think of the Gulf as it gets warm every summer. It's always done that. But what the coral record is showing us is most of the warming is occurring in the summers, not in the winters. The other thing we see is the variability tends to be stronger in the winter time versus the summer. So in the winter, about 3 to 5 degrees Celsius, and in the summer times, it's about 3.5 to 2.5 degrees Celsius variability. So this is just eyeballing the two. We can look at this another way. So what I've done here is plotted a running standard deviation. So for the two records. So here this nicely shows that the summers have lower standard deviation than the winters, which are in blue. So, and we also see that this variability is decreasing as we move from the 1700s into present. So again, these are very small trends in both records, but we do see a decrease in variability within the two, with winters being more variable. Okay. So that's what we see when we look at the seasonality. So what does all this mean? So one of the things I started looking at, this was a recent paper that was published, looking at decadal scale trends in the Gulf of Mexico, the southern part of the Gulf of Mexico and the Caribbean, using very high resolution satellite data. So they have 4-kilometer resolution. Our study site is here with the black star. So, and what this record found using the very high resolution data is that we have these trends varying throughout the Caribbean and the Gulf of Mexico. The trends largest within the Caribbean Sea and the central part of the Gulf of Mexico, but as we move to Florida and the Keys and the Bahamas, these trends become quite low. Now the bottom two graphs show us the error on these trends and the significance of the trends. So you'll notice here in the central part of Gulf of Mexico, we have more variability than we do in the Yucatan area. And if we look at the correlation, as we move into the Bahamas and the Gulf of Mexico, the Gulf of Mexico around Florida, that significance of those decadal trends starts to become quite high. So this tells us these trends are not significant. So how do we interpret that? So this is just 25 years of data. So for the trend to be significant, we need to have more data. So they're limited to 25 years. So this is the noisier part of the Caribbean and Gulf of Mexico when we're looking at trends. And our Dry Tortuga site is right here on the edge of where we move from not being significant to being significant. So if we compare the trend in this high resolution study to this trend we see in the coral records, they do agree. So we see that same 1 degree trend over the 25 years. So the record, or at least the trends I'm seeing in the coral record and what's being shown in this high resolution study, they're showing us that we're seeing about the same things. And then the coral record's allowing us to extend this back further in time. So the next thing I wanted to do was to go through the record and do some bandpass filtering. So where is the variability in the record? So if we look at the different bands, so the top is the interannual band, so 24 months to 10 years or 120 months. Then decadal band from 120 months to 240 months, and then greater than 24 months. If we look at the amount of variance below 24 months or 2 years, that's 95.5% of all the variance in the record. Okay. The interannual band, outside this annual band, has the largest percentage of variance, so 1.65. Our decadal band has very little variance. And then our bidecadal band has 1.86. So I've also plotted this with the black line as the master record, but also with the different cores. So I wanted to see between the different cores, do I have agreement when I start filtering it out for the different bands? So in the interannual band, we have good agreement between the cores. And in the decadal band, similar agreement. and same here in the bidecadal bands with some mean shifts that occur between the two records. Again, we saw that when we were comparing the reproducibility in the beginning. But the variations are moving in the same direction. Okay. So we see we have most of our variances is annual, interannual, and bidecadal. So another way we can look at this is with a wavelet plot. So wavelets is another way to look at a spectrum. But here what we're going to do is plot the spectrum in time space. So what we have on the x-axis here is our time, and on the y-axis is periodicity. So on a typical spectrum, this would be the x-axis. The colors here represent the strength of those different periodicities, and that's shown as a color. So I've also plotted here the coral record, and here I've removed the centennial scale trends. I've plotted the number of cores at the top for your reference. And when we look at the wavelets, we're seeing here all the higher colors tell us we have higher peaks in the spectrum for these time intervals, and where we don't see the blue and the green colors, that tells us we have low spectral peaks. Okay? So in this diagram, everything that has a black line encircling it, these are areas where the concentration of variance is now above the 25% confidence interval. So these would be areas in periodicity time space we could interpret it as being significant. However, there's one caveat to doing wavelet analysis. So you'll notice there's this kind of bowl shape here. So that's called the cone of influence. To do this type of analysis, you have to pad the time series with zeros on the ends. So this produces this cone of influence. So everything that's under the cone of influence, you cannot interpret because of that zero padding. So here where I have significant periodicities, it's being sliced in half with this cone of influence. So I don't have a lot of confidence in this particular area here or in this concentration of variance here. So pretty much what the wavelets is showing me is I don't have any significant concentrations of variance in any of my periodicities. The other thing to consider is here I have two cores, here I have a single core. That's where the significant periodicities are showing up. So if I remove that part of the record and look at the wavelets for just the interval where I have two cores, again, here we see some of the interannual variability starts to come in to being significant, but in the decadal and multi-decadal bands, I still don't have any significant concentrations of variance.
So the red doesn't give you that variance? So like the red patterning at 11 years, like sunspots, wouldn't...
It's not significant.
Okay.
It's not significant at 95%. So. Okay. All right. So to summarize, how am I doing on time? Okay. So we have multiple coral colonies, live and dead. So, and I've shown here that we can extend a coral reconstruction using dead corals. So I've also shown that the gridded SST databases should be used with caution for doing calibration. A lot of coral reconstructions, other types of reconstructions use these data sets as you get back farther in time. So I would suggest for each grid point to evaluate those temperatures to make sure they are robust. I've also shown that the winter temperatures tend to be more variable than the summer. And we see shifts in the sea surface temperature. And based on the location of the dry tortugas, I think these changes are related to changes in the loop current and the Florida current. So I already mentioned the interannual variability. There's no correlation with El Nino. There's no correlation with the AMO. So we don't see that, we don't see either of those in this record. So what I think we're seeing is that variability from the loop current. So looking at the high resolution data, that's telling us this is a very noisy part of the ocean for looking at trends. So what are some of the implications of this study? So one is we're seeing that throughout this 274 years that these summers are warming. So if we have summers are warming, that's a particular concern for coral reefs because this is going to lead to thermal stress, and we also have problems with coral bleaching. So as the water gets warmer and warmer, corals tend to bleach. So we also have with increasing summer temperatures, this can lead to more or stronger hurricanes within the Gulf of Mexico. So those are other things to consider. So another thing is increasing temperatures are linked to moisture flux, particularly into central North America and Florida. So this is some work that Jason Polk up at USF Tampa has been working on. I think Ethan ran most of this data for him in the Paleo Lab. So we've been comparing our two records. So here I'm showing in black, this is the coral reconstruction, plotted a little different. This is a mean annual. And then with Jason's speleothem record from Brooksville, Florida. So that's the black dot right here, dry tortugas down here. So what's interesting is between the records, now Jason's record in this interval has 4 to 5 year resolution. He uses uranium thorium dates as well. He has one date down in this interval. So everything in between is interpolated. But those general trends that we're seeing within the two records are tracking each other. So this is something we expect. So if we look at the top graph here, this is a correlation map of sea surface temperatures in the dry tortugas correlated with precipitation. So whenever we have temperature anomalies here in dry tortugas, we can expect a response in Florida and along the east coast of the U.S., but also in Central America, then up through the Midwest of the U.S. So these anomalies are connected to precipitation anomalies. So that's another interesting aspect that my research and collaborations with Jason we're going to be expanding on. So I'll end it there and take any questions. Yes?
How would the bleaching episode show up in reports?
Good question. There's several studies from the Great Barrier Reef. They've shown that when there's a bleaching event, what happens in the strontium calcium of the coral is it'll shift to a higher ratio. So if a bleaching event had occurred in these corals, we should see that increase. We don't see any in this record, so I don't think these particular corals have bleached, or at least in the way that we could see it. So, so does that answer your question? Okay. Other questions? Yes.
So what do you think is driving the higher variability just higher
In the winters? Well, this goes back to the loop current. So as the loop current comes from the Yucatan into the Gulf of Mexico and then back out, in the summertime when the water's really warm, it's really hard to tell the difference between the loop current and just the waters in the Gulf of Mexico. But in the wintertime, that temperature difference is larger. So you have warmer waters coming from the Caribbean, colder waters in the Gulf of Mexico. So as the loop current moves over the Tortugas platform or close to it and back out, you're going to get that shift between colder waters and warmer waters. So I think that's what's driving the winter variability. So we're seeing that shift in the loop current. So I know there's experts in the room on the loop current. So any thoughts you have, I am very open to any thoughts you may have.
You see anything different or unusual about the years 1983 and 84?
Not in these corals. So nothing that looks really unusual in them. So yeah, that was one of the things we were concerned about if there was bleaching events or the El Nino showing up.
Start dying all over the Caribbean.
Right. Yeah, I didn't put the plots up in this presentation. I have run the extinction rates in all of the corals based on their chemistry. So the extinction rates are all different between all four of the corals. So one of them's increasing, one's decreasing, but I don't see anything from the growth or the linear extension that's telling me there's a decline in any particular, or a decline that's occurring over all four corals. So, a good question, Gene. So, yes, David.
So, you were saying about the summer temperatures. Again, coming back to the extension rate, can you do them on a seasonal basis rather than on an annual basis? And is there no relationships between growth and temperature?
Yes, I've looked at that. I don't see, if I do just the paths chemistry with the extinction rate per path, there's no significant correlations. As I start to average everything together, so some parts of the record I have 4 and 5 paths that I'm averaging together. In the core from the the live top of the largest colony, that one has a 0.22 correlation between average growth and average strontium calcium. The other ones, no significant correlation. And they're all going in different directions.
This is all Pseudoraster.
Yes, yes.
You
So-
might see it monash.
Right, in another species we might see something different. So in these, I don't see any correlation between growth rate and strontium calcium. Another thing I looked at was what is that lower growth limit that I can get to before I see a shift in the chemistry. So our lowest growth rates in this record are 0.2, and at 0.2 to 0.3, I don't see any shifts in the strontium calcium. So there could be shifts with lower growth rates, I just don't have them in this coral.
If you go back 2 slides, what happened between 1770 and
This one?
1790?
1770 and 1790?
Yeah.
I know, it's a big shift. So this is in here. Let me go
I mean,
back.
you see, it's in the central Gulf of Mexico, right? Julie?
Right in here?
I don't know if I can resolve the timing.
Yeah. This is right where I'm moving from 2 cores to a single core. So this is one where if I had a second core here where I could replicate it, I would be a little more confident in this shift. But
Jason does the same thing.
we see it in Jason's record and we see it in Julie's record. So,
So say something.
so something's happening here, and it's a big shift. So there's also a lady, Sherry Johnson. She's in Georgia. She's doing historical climatology down in Cuba. She sees a big shift in Cuba in this interval in her historical records as well. So something's happening in here where we have a shift to colder temperatures. And this
winter
is
summer compare in that, although albeit 1 core.
right.
How does winter summer compare there?
One summer.
I mean, just a win, you know, the sort of, I guess you can bin them in the 1780 to 1795, and
Right.
Is there any summer winter variability that happens there?
Let's see. So this is right in here. So we see summers are dropping. The winters is a little bit of drop, but not a lot. So it's mostly in the summers where it's occurring. So Sherry's talked about in hers an extended El Nino. I don't know if I quite buy that. So, but something's happening. So I think with Julie's record and Jason's record, I mean, timing-wise, they all look like they're lining up, but there's error bars on their timing too. So, yeah.
On that same event, if you just continue that 1 core forward without averaging, does it have to drop? Go back to the previous one.
Mm-hmm.
See where it transitions in 1780
Okay.
from 1 to 2.
We can do it. I'll go back to that. So this is, sorry. So that would be right in here, that shift. So there is a small shift there.
No, it's later.
So it's like 1780.
It's later.
Right.
Anyway, we can look at it later.
Right. So yeah, it's one of those things. It's where you're splicing 2 records together. So there's little shifts that occur, but there's a little drop there. So something happened there, Dave?
No, I'm.
Oh, okay. Well, there's no big volcanic events that I know of at that time. So that's in the 1810s where you get Tambora.
Central Caribbean or North Caribbean Cariaco record,
I haven't looked at that yet. I should do that. So,
or the lake records from Mexico.
right. So,
Yeah, from that.
yeah. So there,
Awesome.
yeah, I'm still exploring all the different teleconnections and so what does all of this mean, and so
Didn't you do that, Julie?
yeah. Well, I think with Julie's we were thinking it was AMO, so type variability and
I mean, I can't resolve the timing of an event like that.
right,
Uncertainties in radiocarbon dating.
right. So
You know,
yes, Steve.
Elizabeth Merman, in
Mhm.
her master's she did here, she found the Krakatoa dust in 1583
Right.
and a little afterwards.
Mhm.
And, don't see any effect.
Yeah, the Krakatoa and Tambora both show up So that's this cooling event right here, it's Tambora, and the Krakatoa is this one right here. So the cooling shows up from both of those large eruptions. So, yeah, meteorite.
Anything unusual about 1942?
1942, middle World War II.
There's a major stress band in every
Right.
coral we cord, and the keys hit that date.
Right.
It never correlated with anything.
Right. I'll have to go back and look in the SIDS and see if we see anything. The SIDS don't have the nice density bands that the Montastrias do. They're kind of blurry. So, but one of the things that's kind of cool, I'll jump back. Oops, sorry, I'm going the wrong direction. When I was lining up the two corals, the two cores, and doing the band counting, so the bands are really tiny in these two X-rays, but you'll notice here here you have dark bands, light bands, dark bands. These are all like 10-year bands. These bands are consistent between the colony, between the two cores. So I use those bands to help align these cores. So that was something
What do they mean?
yeah, it's consistent throughout the colony, and they do line up. So it's something within the colony. You're having years where it's putting down denser skeleton for 5 to 10 years, and then it shifts to lighter skeleton. So that's some of the things I've noticed looking at that. So I've kind of played with lining this core up with this one to see if those light dark bands match up. This X-ray here, we need to do a little better. So this was all film X-ray. This one we did with the high resolution X-ray at LSU. So I'll run this one through the high resolution X-ray and get a little better X-ray, and then we might be able to line up those density band patterns. What they are, I don't know. Ilsa, maybe. I don't know if you've seen them before. So.
There's a lot of mysterious
Mm-hmm.
bands. Yeah, I don't know.
Right. But yeah, there's definitely band couplets within the colony.
How long do the light bands last relative to the dark ones?
It's like 5 to 10 years.
In both cases?
Mm-hmm. Yeah, it just switches back and forth between it. Why? I don't see it in parietes.
Decadal oscillation.
Maybe in coral growth. I don't know. So,
Is it just density that's causing the color, or can you ever do any scanning for other elements? I know you guys are fixed by methodology on your
right.
Have you broadened the spectrum to see what else is in there?
So, like, as far as like magnesium, calcium, or oxygen
Forget
isotopes? Right.
Whatever other trace element there may be.
We're running the oxygen isotopes. Chris actually sent me the top part of the F1 coral a couple weeks ago. The oxygen isotopes between the A1 and F1 coral line up nicely with each other.
I was thinking more exotic.
More exotic? No, we have, well, I take that back. We are doing a little more exotic things. Michelle Levine, who is up at Rutgers, she's run a full suite of elements on the ICP-MS. She did her dissertation work looking at phosphate in corals. So in these particular corals, we didn't see anything phosphate-wise that was too exciting, but the barium is doing something different. So the barium has a perfect seasonal cycle in it, offset from strontium calcium by 1 month. So her hypothesis is that it's chlorophyll driven. So one of the things I'm doing on Monday is I'm going to drill her some more samples that she's going to run. We're going to replicate it within the two corals to see if that barium lines up with a chlorophyll spike that happened in 1998. So we're in the process of doing that. But yeah, the barium has something weird in it. So, but we have done the full suite for about three years, and that's the only one that stood out as being really different. So.
Yeah.
What, with your NDVC data, the Pulaski Shoals and the other dry Portuguese station, the error or the precision of those instruments, the thermistors they use on there is not great. It's something like 0.5 C.
Right.
Within the error that you're showing between your
Right.
two different data sets. So that's
For an
you
hourly
calculate
measurement.
Error on on an hour. You calculate error on a monthly scale.
Correct.
But you're still getting, your RMS is less than the precision of the instrument of
Right.
the NBC instrument?
No, it's a good question. And it's because they're averages. So as you average something, so your standard error of a mean is you take that standard deviation, divide it by the square root of number of observations. So it's going to diminish as you have more observations. So,
Are there any more questions? Oh,
I need to go back further in time. First, two observations. One is that I think the thickest Holocene coral section is in the Dryper-Keys, thicker than anywhere else in the Keys. So I wonder why that is. And then, when did Holocene corals really begin in Florida?
Right.
Because they don't appear back 14,000 years ago. They don't have, and is it strictly sea surface temperature warming up above 20 degrees C so corals could survive?
right. Or sea levels high enough for corals to
Well,
grow?
sea levels, there's always a seafloor, and there's always
Right.
shallow water.
Right.
And there's always enough light for
Right.
corals,
Right,
regardless where you are. But there are no corals.
right.
And so,
Well,
when coming out of the last glacial maximum,
Right.
when did the Holocene or
Right.
Pleistocene slash
Right.
early Holocene reef get developed?
Right. Well, Harry Robbins, who's at LSU, he published recently finding some Holocene corals in deeper water off the coast of Louisiana. So when I get back, I'm going to go over to his lab and look at some of his cores and talk to him about those. So he has some off the coast of Louisiana, and then also some researchers at Rice have found some deeper water corals that look to be Holocene off the Texas coast and Mexican coast. So I think they're out there, it's just finding them. So,
Well, all this area you're looking at here, it was not flooded until between 6,000 and
Right.
7,000 years ago.
right,
Once you go further down slope,
right,
there's shallow water.
right,
So these could have formed down there, but they didn't form.
right. So, no, I think it's all interesting questions, so,
The bottom of the reef tract off the east coast of Florida dates at like 9,000 years. And there's corals there. And there's definitely some by 7,000.
Right. So when did he get cranked up? I guess that's my question.
Right.
And what triggered it to get cranked up?
right. No, I think they're good questions, so I don't know the answers, so.
Would you not find synchronicity with other provenances globally at that point, if it's a sea level relationship? Or there's some key, no key.
Right.
I
Well,
don't know when the Great Barrier Reef got cranked up.
well, it's about that same time interval. So I think for sea level to stabilize, so if you have lots of fluctuations, it's hard for corals to grow, so you need sea level to stabilize. Also in the Gulf of Mexico, we've got all that freshwater flux coming off the Laurentide ice sheet and all that sediment coming from the glaciers. So that's not going to be conducive to corals growing. So I think there's lots of different factors that could play into it. So, but yeah, I would love to go out and find them if they're out there. So,
are there any more questions? I guess we'll conclude, and thank you, Kristine, for coming.
thanks.